The human microbiome is the collection of trillions of microorganisms living on and inside the human body. The microbiome includes various types of microorganisms. Bacteria are the most numerous, but fungi, viruses, and other microbes also play important roles. These microorganisms are mostly beneficial and essential for human health. They aid in digestion and nutrient absorption, train our immune system, and protect us from harmful pathogens. The scale of the microbiome is impressive. Microbial cells outnumber human cells by a ratio of about 10 to 1. Together, they weigh about 2 kilograms, roughly the weight of a human brain. The microbiome is distributed throughout the body, with distinct communities in different locations. The skin hosts a diverse community adapted to different environmental conditions. The oral cavity and respiratory tract have their own unique populations. The gut contains the largest and most diverse microbial community. The urogenital tract maintains specialized microorganisms adapted to its unique environment. Our body hosts two main types of normal flora, resident and transient. Normal flora are microorganisms that inhabit our body surfaces without causing disease under normal conditions. Let's compare the two main types of flora that live on and in our bodies. Resident flora are permanent inhabitants of the body, while transient flora are temporary visitors. Resident flora are permanent inhabitants that have established themselves in specific body locations. They are well adapted to their environments and maintain stable populations. Transient flora, in contrast, are temporary visitors that may be present for hours, days, or weeks before being removed. Their populations fluctuate significantly. Resident flora provide long-term benefits like preventing pathogen colonization and supporting immune function. Transient flora may be beneficial, neutral, or potentially harmful depending on circumstances. Let's look at some examples of where we find resident and transient flora in the human body. Resident flora includes Staphylococcus epidermidis on the skin, Lactobacillus in the gut, and Streptococcus in the mouth. Transient flora may include E. coli temporarily on hands, seasonal respiratory microbes, and food-associated bacteria passing through the gut. Both resident and transient flora play important roles in our health. Resident flora provide colonization resistance by occupying niches that might otherwise be filled by pathogens. They also help train our immune system from birth. Transient flora introduce microbial diversity and can bring new beneficial functions like additional digestive capabilities. The balance between both types is key to maintaining health. The human skin is home to diverse microbial communities that play crucial roles in maintaining skin health. Different skin regions harbor distinct microbial populations based on the local environment conditions. The composition of skin microbiota is influenced by several factors, including moisture levels, pH, sebum production, and temperature. Let's take a closer look at the key bacterial species that dominate the skin microbiome. Staphylococcus epidermidis is the most abundant bacterium on the skin surface, playing a crucial role in maintaining skin health. Corynebacteria thrive in moist areas such as the armpit and groin, contributing to body odor through their metabolic activities. Propionibacteria dominate sebaceous regions like the face and scalp, where they break down sebum into free fatty acids that help maintain skin pH. The skin microbiome serves several important functions that benefit our skin and overall health. Let's examine a cross-section of skin to better understand where different microbial communities reside. These microbes interact with skin cells in various ways, forming a complex ecosystem. Understanding the skin microbiome helps us appreciate how these microbial communities contribute to skin health and immunity. Section 4. Oral and respiratory microbiomes. The human body hosts complex microbial communities in both the oral cavity and respiratory tract. The oral microbiome is one of the most diverse microbial communities in the human body, containing hundreds of bacterial species that form complex ecosystems. Key species include Streptococcus mutans, which forms dental plaque, and Porphyromonas, which is associated with gum disease. These and other bacteria have adapted to specific niches within the mouth. These bacteria form dental biofilms through a multi-stage process. First, bacteria attach to the tooth surface, then they multiply to form microcolonies. Finally, a mature biofilm develops with a protective matrix that shields the bacteria. In contrast to the oral cavity, the respiratory tract shows a gradient of microbial colonization. The upper respiratory tract, including the nasal passages and throat, contains various microbes. However, the lower respiratory tract is nearly sterile in healthy individuals with only minimal microbial presence. Throughout both the oral cavity and respiratory tract, microbes interact with mucous membranes. These microbes colonize the mucus layer covering epithelial cells, forming a protective barrier and interacting with the host immune system. One of the most important functions of the normal flora in these regions is preventing infection through competitive exclusion. Resident microbes occupy attachment sites, produce antimicrobial substances, and compete for nutrients, thereby preventing colonization by respiratory pathogens. The balance of microbes in the oral and respiratory microbiomes is crucial for health. Disruption of these communities can lead to conditions like dental caries, periodontal disease, or respiratory infections. Let's explore the gut microbiome and the differences between the small and large intestines. The gut microbiome varies dramatically throughout the digestive tract, with a gradient of microbial populations from the stomach to the colon. The small intestine has relatively few microbes due to rapid transit time and antimicrobial secretions like bile. Bacterial density here ranges from 10 quip to 10 fret colony-forming units per milliliter, predominantly firmicutes and proteobacteria phyla. The environment is less hospitable to microbes due to rapid transit time and antimicrobial bile salts and digestive enzymes. This area focuses on nutrient absorption rather than microbial fermentation. In contrast, the large intestine contains the highest microbial density in the human body. Bacterial counts reach an astounding 10 to 10 squared colony-forming units per milliliter, primarily dominated by firmicutes and Bacteroidetes phyla. These large intestinal microbes perform critical functions like breaking down complex carbohydrates that our digestive enzymes cannot process. Through fermentation, they produce short-chain fatty acids like butyrate, acetate, and propionate, which nourish intestinal cells and regulate metabolism. These gut microbes continuously interact with intestinal cells, influencing immune function and maintaining the intestinal barrier. We can observe a clear gradient of microbial populations as we move from the stomach through the small intestine and into the large intestine. Bacterial density increases dramatically from approximately 10 thin to 10 wqi in the stomach, reaching 10 to 10 fites in the terminal ileum and culminating at 10 to 10 quaid in the colon. To summarize the key differences, the small intestine has fewer microbes, different dominant bacterial phyla, and focuses on nutrient absorption. The large intestine has much higher bacterial density, longer transit time, and specializes in fermentation of complex carbohydrates, producing beneficial metabolites for the host. Specialized microbiomes exist throughout the body, adapting to unique local environments and providing tailored protection. The vaginal microbiome is predominantly composed of lactobacillus species. These beneficial bacteria are critical for maintaining vaginal health. Lactobacilli produce lactic acid, which maintains the vaginal pH between 3.8 and 4.5. This acidic environment prevents the growth of pathogenic bacteria. The vaginal microbiome composition changes during the menstrual cycle and pregnancy, but consistently provides a crucial barrier against infections. Unlike most body sites, the urinary tract is typically sterile above the urethra, meaning it normally contains no microorganisms. The sterility is maintained by regular urine flow, which flushes away potential pathogens. Only the urethral entrance may harbor some resident microbes. This is a stark contrast to the vaginal microbiome, which has a high bacterial load and acidic pH, while the urinary tract has very few microbes and a more neutral pH. The ear canal supports a sparse but specialized microbiome. Unlike internal ear structures, which are typically sterile, the ear canal harbors a small community of microorganisms. This microbiome is primarily dominated by Staphylococcus and Corynebacterium species. Cerumen, commonly known as earwax, contains antimicrobial compounds that help regulate the microbial population and maintain ear canal health. The conjunctiva of the eye supports an extremely sparse microbiome. This environment is continuously cleared by blinking, which mechanically removes microorganisms. The tear film contains powerful antimicrobial compounds like lysozyme, which destroys bacterial cell walls, and lactoferrin, which sequesters iron needed for bacterial growth. These adaptations keep the eye surface nearly sterile. Each specialized microbiome has evolved unique adaptations to its local environment. The vaginal microbiome maintains an acidic pH through lactobacillus activity. The urinary tract remains nearly sterile due to regular urine flow. The ear canal is protected by antimicrobial compounds in cerumen, and the eye conjunctiva is defended by powerful enzymes in tears. These microbiomes have evolved specific defense mechanisms adapted to their unique anatomical locations and physiological conditions. Various factors shape the composition of our microbiome, impacting which organisms thrive and which decline. Age affects which microbes populate our bodies from birth through adulthood to old age. Diet has one of the strongest impacts, especially on gut microbiome composition. Hormonal changes during puberty, pregnancy, and menopause alter microbial communities. Hygiene practices like hand washing and bathing influence skin and mucosal microbiomes. Antibiotic use can dramatically disrupt normal microbial balance, often with lasting effects. Immune system status determines which microbes are tolerated versus eliminated. Environmental exposures, including location, animals, and plants, introduce new microbes. Genetic factors influence which microbes can establish themselves in our bodies. Let's take a closer look at how diet impacts our microbiome. Diet has a particularly strong influence on gut microbiome composition, with different food groups promoting different bacterial communities. A diet rich in plant fibers promotes beneficial bacteria like Bacteroidetes and Bifidobacteria, while high-protein diets favor different communities like Proteobacteria. Our microbiome also changes throughout our lifetime. Our microbiome evolves throughout our lifespan, beginning at birth and continuing to change as we age. Newborns start with limited microbial diversity, which rapidly increases during infancy and childhood. Diversity peaks in adulthood before decreasing in old age. Antibiotics can dramatically disrupt our microbiome balance. Before antibiotic treatment, our microbiome exists in a balanced state with diverse, beneficial bacteria. During antibiotic treatment, both harmful and beneficial bacteria are significantly reduced. In the recovery phase, bacteria begin to repopulate, but the balance may be temporarily disrupted. Eventually, a new equilibrium is established, though it may differ from the original microbiome composition. Multiple factors continuously shape our microbiome throughout life, from diet and age to antibiotics and genetics, creating our unique microbial fingerprint. Understanding these influences helps us maintain and restore microbiome health. Normal flora, the microorganisms that typically inhabit our bodies, can become harmful under certain conditions. These are called opportunistic pathogens, normally harmless microbes that take advantage of opportunities to cause disease. One way normal flora becomes harmful is by entering the wrong body location. A classic example is urinary tract infections. Escherichia coli, or E. coli, normally lives harmlessly in our intestines. But if these bacteria translocate to the urinary tract, they can cause infection and inflammation. Another way normal flora becomes harmful is when host defenses are compromised. In the oral cavity, we normally maintain a balanced microbial community. When factors like high sugar diet, poor oral hygiene, or reduced saliva flow compromise our defenses, bacteria like Streptococcus mutans can overgrow and cause dental caries. This shift from healthy microbiome to disease-causing state is called dysbiosis, a microbial imbalance that contributes to various diseases. In a balanced microbiome, beneficial and commensal bacteria dominate, with potential pathogens kept in check. Various triggers like antibiotics, diet changes, inflammation, or stress can disrupt this balance, leading to dysbiosis where potential pathogens overgrow. One of the most dramatic examples of normal flora becoming harmful is Clostridium difficile infection. In a healthy gut, a diverse microbiome keeps potential pathogens like C. difficile in check through competition. Antibiotic treatment, while targeting pathogens, also eliminates beneficial bacteria that normally compete with C. difficile. This creates an opportunity for C. difficile to multiply rapidly, causing severe diarrhea, inflammation, and potentially life-threatening complications. Maintaining a healthy microbiome is crucial for overall health. Probiotics are live microorganisms that provide health benefits when consumed in adequate amounts. They help restore and maintain beneficial bacteria in the gut. Common sources of probiotics include yogurt, kimchi, kefir, and kombucha. Prebiotics are specialized plant fibers that act as food for beneficial bacteria. Unlike probiotics, prebiotics aren't living organisms, but rather nutrients that help existing good bacteria thrive. When prebiotic fiber reaches the gut, it serves as nourishment for beneficial bacteria, helping them multiply and produce helpful compounds. Prebiotic foods include garlic, onions, bananas, oats, apples, and flax seeds. Fecal microbiota transplantation, or FMT, is a procedure that transfers fecal bacteria from a healthy donor to restore the gut microbiome of a recipient. The process involves collecting stool from a carefully screened, healthy donor, processing it in a laboratory, and then delivering it to the recipient through various methods such as colonoscopy, enema, or capsules. FMT is primarily used to treat recurrent Clostridium difficile infections, with success rates of around 90% after 1 or 2 treatments. There are several practical ways to maintain a healthy microbiome in everyday life. Eating a diverse plant-based diet rich in fiber provides fuel for beneficial bacteria. Limiting unnecessary antibiotic use helps preserve your existing microbial communities. Regular physical activity has been shown to increase microbial diversity. Managing stress is important, as stress hormones can alter gut bacteria composition. Quality sleep is also essential, as disrupted sleep patterns can negatively impact the microbiome. Looking to the future, scientists are developing targeted microbiome-based therapies for various health conditions. For inflammatory bowel disease, microbiome-based treatments show significant promise, with clinical trials already underway. Research on addressing obesity and metabolic disorders through microbiome manipulation is progressing rapidly. The gut-brain axis connection has opened new avenues for treating mental health conditions through the microbiome. Early research suggests the microbiome may also influence cancer treatment responses, particularly in immunotherapy. Maintaining microbiome health requires both preventive measures and targeted interventions. As research advances, personalized microbiome approaches may become standard in healthcare.